Hello everyone, welcome back to VAV's webinar series 2020. I'm Janvi, your host for today, and I thank you all for registering for another session. So in our previous session, we have known how to formulate liposomes and what is the setup required, equipments required to design liposomes. So moving on from here, we will now learn how to select the right grade of lipids for the right type of liposomes for the intended application. Along with the selection of lipids, it is also important to emphasize on the crucial or the critical raw materials required. So let's welcome our speaker for today, Mr. Arun Kedia, to explain the factors influencing the selection of lipids and other crucial raw materials. Mr. Kedia is a chemical engineer by education. He is the founder director of VAV Group, which is established in 2003, and he has dedicated 3 decades to the research and to understand the unique aspects of phospholipids. Here's presenting Mr. Kedia.
Thank you, Janvi, for the introduction, and good afternoon, everybody, or good morning, depending on your geography. So today we will talk about primarily the selection of lipids, and this has been a challenge for a lot of researchers, limited understanding of lipids, and often leading to project failures or delays or instability topics. So let's begin. So today we will quickly recap the introduction to liposomes, but this was already covered in the previous seminars, the role of phospholipids, the critical factors in lipid selection, and what other materials can have a role or impact on liposomes. So just to quickly recap, liposomes are artificially prepared spherical vesicles derived from phospholipids, can combine other lipids. They have an aqueous core, and they can virtually administer any active. And as you see from the picture, the hydrophilic payload sits in the center, and the lipid bilayer can accommodate the hydrophobic molecules. So phospholipids will form liposomes spontaneously on hydration. This is a statement which you hear quite often enough. Except there is a condition: only when the concentration of lipids is greater than the critical micellar concentration. Now the CMC, or the critical micellar concentration, fortunately runs in very low concentrations, usually in the space of millimoles, and only when this concentration, the CMC, is exceeded, then the phospholipids start to agglomerate as vesicles. Also to highlight here that the CMC can depend on the lipid, and so it's like a physical property of the lipid. So let's look at the critical factors in lipid selection. Finally, the objective being agreed is to design stable, efficacious, consistent liposome systems. And good liposomes, or well-designed liposomes, will have a positive effect on membrane characteristics like permeability, lateral diffusion of lipids, elasticity, binding properties, drug loading, etc. So the first question always in lipid selection: natural or synthetic? We will touch upon this in great detail. Natural grades depend on, the grades can widely depend on the composition, the source, and consequently the stability on the grade. Now, the more classical natural lipids used in liposomes is soya bean, most common, sunflower, these are from plant sources, And then we can have within the animal kingdom, most common is egg sources. Then the second choice can be synthetic lipid. Now, when we say synthetic, these are also nature derived molecules, but with modification, and they are usually engineered for a desired release profile or a stability profile. Uh, Important to note here, when we talk of unsaturation of the fatty acids, then the synthetics are the most saturated, followed by egg, followed by plant. Plant sources are usually the most unsaturated. Uh, an important factor in liposome design is the final commercialization. So natural lipids are relatively economical, and the good news is that they are adequate for most applications. So you should only look at synthetic sources once you have eliminated the possibility of using a natural lipid. Now, we talk of pH sensitivity, and there are two kinds of pH sensitivity. So, first is the pH sensitivity during the formulation process. Now, liposomes are sensitive to extremes of pH. So, we explain it in detail in subsequent slides, but deionized ultra pure water is important for the stability of the liposome on storage. And then when you are encapsulating drugs of extreme pH, which are strongly ionizing, then always suitable buffers must be used to maintain the pH within an acceptable range. And thirdly, the presence of free fatty acids in lipids. Now, these can appear as impurities, uh, and they are extremely detrimental to liposome stability when we are talking of uh, pH sensitivity during formulation. Then the next interesting question is what happens in vivo. Now, in vivo, the pH sensitivity is actually an interesting drug delivery question. So, rapid degradation of pH sensitive liposomes occurs by lysosomal enzymes, and specific lipids having titrable head groups are usually added to form stable but pH sensitive liposomes, which means they will trigger release when they find an altered pH, but they are generally otherwise quite stable. And it's like an important design factor. So you see in the graph that you can have an altered pH within the tumor cells compared to the healthy cells adjoining, and so the liposome can release when it finds the altered pH. An extremely important factor in liposome design is the phase transition temperature of lipids. So first of all, what is phase transition, or it's also referred to as the glass transition temperature? So it's the temperature when phospholipids will transit from gel phase to the liquid crystalline phase. Erroneously, it is sometimes referred to as similar to melting point. It's nothing similar to melting point. And what are the factors? This depends on the PTT. It depends on the carbon chain length, degree of unsaturation of fatty acids, and the polar head. Just to illustrate the last point, so within PC and PG with identical fatty acids, PTT is similar. But if you look at the same fatty acid chain lengths, then they can be much higher for P. So the polar head has a role. And typically the phase transition temperature of plant sources is lower than egg sources. And within synthetics, if we look at homogeneous saturated phospholipids, of course, synthetics can have a wide range of phospholipids, but we've taken or we picked 4 of the most common. So DLPC will be less than DMPC, less than DPPC. So basically, it's increasing the chain length with the same degree of fully saturation. It's simply a function of the higher the chain length, higher is the PTT. And you can very cleverly design or use this feature to design or engineer your release profile. Also important to note that all natural phospholipids will typically, because of the mixture of fatty acids and the mixture of polar heads, have quite a wide range of PTT. As opposed to synthetic phospholipids, it's identical fatty acids, always the same polar head. So they will have a very sharp PTT. Do we always need sharp PTT? You can decide based on the nature of your formulation. Now another look at, okay, so what is the significance of phase transition? So in the graph or in the picture, you see that the both the phases, the gel phase where the fatty acids are quite rigid, and this is at usually low temperatures lower than the PTT. And then as you exceed the PTT, you will have the liquid crystalline phase, and if you observe, the fatty acids are much more fluid or in a more dynamic state. And what is the impact? So you can have a non-leaky vesicle produced with a high phase transition lipid. The vesicle is always in the gel phase at storage temperature, and preferably in the liquid crystalline phase during release. But this is a debatable statement. So if the liposome also continues to be in gel phase in vivo, then you will have a much slower or a prolonged release or a sustained release. And then obviously, if you look at the fatty acids, the vesicles become leaky when the lipid passes through its PTT to the liquid crystalline phase, rapidly releasing the encapsulated active. Now, choice of lipids specific to injectable liposomes. So the mononuclear phagocyte system, MPS, is able to identify liposomes based on the composition. This is a very long topic and probably suitable for a dedicated webinar. So I'll not delve too deep. But so liposomes can be resistant to clearance when they resemble the composition of exterior layer of RBCs. And the stealth properties can be enhanced by pegylation, polyethylene glycol. Then the membrane fluidity also affects the clearance of liposomes. So, for example, long circulating liposomes can be prepared for transporting drugs to the sites of diseases beyond the MPS so as to overcome the rapid elimination. And further, you can reduce the MPS clearance by enhancing the rigidity of the liposomes by adding either phospholipids with high PTT or cholesterol or sphingomyelins. Sphingomyelins being also naturally present in egg phospholipids. Then the question of stability. So in general, saturated lipids are more stable to oxidation. But be careful here because they are more vulnerable to hydrolysis. And so quite often the question asked by researchers is that should I only start with synthetics because they are fully saturated? My answer is not necessarily. There are other factors to consider because even if you have oxidation stability, you can have the risk of hydrolysis, and all liposomes will have water. Now, what makes let's say the natural phospholipids more stable to hydrolysis, relatively speaking? So carbohydrates of natural lipids will displace water from the membrane water interface by intercalating into the head group region near this interface, which means just outside the shell. And this stabilizes the membrane to hydrolysis from the bulk water phase. Of course, there can be other factors like temperature, pH, acyl chain length, head group, ionic strength, and the strength of aggregation. These will all be factors which can impact hydrolysis. Another important factor to consider charge. Uh, be careful, we are referring to the charge of the liposome here and not the zeta potential of the solution or the emulsion. So in general, anionic phospholipids in biological membranes will impart a net negative charge. And you see some examples: PS, PI, PA, cardiolipin. These are major anionic phospholipids occurring naturally. So this negative charge impacts or imparts special functions to the membranes, uh, things like adhesion, things like the clearance, etc. And both charge and specificity for particular cells must be considered while designing the liposomes. So just to continue, so the composition of complex lipid mixtures is basically designed to mimic the biological function, optimize the charge ratio, uh, look at the unsaturation, so which has an impact on the phase transition, and finally all of these will have an impact on the release and efficacy profile. Uh, in general, natural lipids are more suitable for immediate release, and saturated synthetic lipids, homogeneous, are suitable for sustained release. Typically, if you use synthetic lipids like DPPC or DSPC, DSPC having a extremely high phase transition, so it always remains in the gel phase, even inside the body. And so because it's not crossing the phase transition, only the the enzymatic action will degrade the liposome, which is a slow action, and so you will have a slower or a sustained release effect. uh, and in lipid mixtures, be careful that if you make a lipid mixture, so let's say if you mixed DPPC with DSPC, uh, and both have different phase transition, then the liposomes will start releasing above the temperature of the lower PTT lipid. So which means as soon as you cross the DPPC phase transition temperature, you will have fluidity of the fatty acids of the DPPC, and so the release will start. But this is a quite an interesting topic, because if you used, depending on the composition you use for or the mix of DPPC and DSPC, you can actually control the release profile. So the Cmax, the Tmax, the AUC, all of these can be actually specifically designed for the desirable release profile. So moving on, what are the other materials which can be critical to liposome design besides just the choice of phospholipids? It's and you might find this humorous, but probably the single most critical factor after the choice of lipid is the quality of water. We have seen a lot of failures of projects, typically because not enough attention was paid to the water. And so you must always use ultra pure and deoxygenated water. These limits have to be almost strictly enforced. So extremely high resistivity or very low conductivity, resistivity typically being in the 15 mega ohms space. The total organic carbon, which is an indication of organic impurities, should be quite low. The dissolved oxygen almost absent, and then you can choose your microbial and BET limits as per water for injection, or this depends on what is the final values you need for the long-term storage. But so it's a factor which we normally cannot emphasize enough: quality of water. Then the quality of the solvent system used. So the solvent purity is quite important, and this is to prevent destabilizing ions. Again, researchers make a common mistake because the solvent is added to dissolve the phospholipid and then finally it will be evaporated. It's not part of the finished formulation. So maybe less attention is paid. But while it's there for whatever short while, it has quite a role. And any impurities which the solvent will contain, even after the solvent is evaporated, the impurities will continue to remain in your formulation. So it's quite an important factor. Uh, the polarity of solvent is important to ensure dissolution of all the lipids, not only phospholipids, but complete dissolution of all the lipids at a molecular level for maximum encapsulation efficiency. If the solvent polarity is not right, then you may have part of the lipids undissolved at a at a molecular level. And then if you subsequently do the microfiltering, these lipids can be lost. Not only are the lipids lost, but then you can alter the original composition because you don't know if the cholesterol was filtered out or what was filtered out. Then the next factor is the cholesterol to lipid ratio. This basically ensures the sturdiness of the liposomes and the rigidity of the membrane. So this ratio should be optimized for leak stability of the liposomes on storage. However, there is a factor which has to be considered. The cholesterol will occupy exactly the same space as your hydrophobic molecules, which is the lipid bilayer. So if you are loading more cholesterol, then you are automatically compromising the space available for your hydrophobic drug. Now, if your hydrophobic drug was a high potency drug, which means you only need a few milligrams to be inside the liposome, probably it doesn't matter. But if you go to high loading drugs such as vitamin C or curcumin, then you need almost or as much as space you can get in the lipid bilayer, and so you must limit the content of cholesterol just enough to ensure membrane rigidity, but not too much. So with that, I will end my presentation, and of course, as usual, open to questions.
Yeah. Thank you, Mr. Kedia, for your precise guidelines for selecting the lipids and the appropriate raw materials. I assume our attendees have noted these guidelines so as to implement while sourcing ingredients for their liposomal projects. We'll now proceed to our Q&A session, and we'll be happy to answer as many queries as time permits. Yes. So first question is, how can we avoid accelerated blood clearance phenomenon by using pegylated liposomes?
Yeah, this is quite a standard technique is to use pegylation to avoid, or I would say not avoid, but to delay the ABC. Which this technique is often referred to also as passive targeting, and this is yeah, you can do it by in two ways. Either you can take the lipid and then carry out the pegylation by yourself, or there are also pegylated lipids readily available, and you can use both. There are papers which talk about increased toxicity by other organs or other cells because of the pegylation, because of the long circulation. So it must be a balance factor. It's not automatically in unidirectional, but it's a valid approach to avoiding the ABC.
Yeah. So can you explain how to choose phospholipids for phytosomes? Can we apply the same logic?
Absolutely. So the basic logic, basic science does not change. If the active is a phytochemical, is it a cytotoxic molecule, it doesn't change. The basic principles in general for phytosomes, so the release profile, the these may not be critical factors. So you don't need the sharp ptt, you don't need sustained release. So the natural choice can be natural lipids. And also for phytosomes, you have always economic considerations. uh, those formulations being usually being used for long term dosage instead of a week. So you will have economic questions also. So the natural phospholipids are ideally suited for phytosomes. And usually it's the plant origin natural phospholipids, so typically soya bean first choice. However, if you have an allergen question or a non GMO question, then sunflower is a valid choice. Nothing to exclude egg phospholipids, but any natural phospholipid can be a good choice. Then, depending on your other factors, you may choose the right grade of the natural phospholipid. This is not only not to be ignored, because within, I mean, for example, our company within soya, I think we have something like 25 grades. So the choice of the grade is also important. Okay, I think I see the next question is how to increase encapsulation of water soluble drugs into liposomes. It's an interesting question. So when we talk of let's say hydrophilic drugs, they will always sit in the core of the liposomes. And if you had a multilamellar liposome or a large liposome, the core area or the core volume will remain constant, which means logically you should have small unilamellar liposomes. And that's the way to increase encapsulation of water soluble drugs into the liposomes. Now, what does this mean? So this topic was covered in greater detail in our previous webinar, but in general you should use good manufacturing techniques to get small unilamellar liposome. That's the way to.
Yeah, so we'll move on to the next one, which is in cases where combination of lipids is used, how do we decide the lipid ratio?
Okay, this was partially covered in my slide. So let's look at the two parts independently. One is the stabilizing lipids, for example, cholesterol, sphingomyelin, etc., and then the phospholipids in themselves. So the stabilizing lipids, typically cholesterol, if you don't know their exact ratio, you could start with something like between 10 and 20%, and then optimize in either direction for your membrane rigidity or the release profile. Now, if we look at the phospholipids, then one factor can be, of course, the phase transition temperature. It can be the combination of lipids, so PC to P to PI to PA. Here, I should point out that most common mistake which researchers make is only to look at phosphatidylcholine as the phospholipid of choice or interest for liposomes. It's a gross mistake. All phospholipids, PCP, PIPA, will form liposomes. And quite often, the combination of lipids can give you more stable liposomes than a single lipid in itself. So, yeah, you can use these factors to design the mix.
Okay. So, how critically the nature of the drug affect the choice of lipids?
Quite important, quite critically. So, if first of all, your first question is what is the nature of the drug in terms of hydrophobicity or hydrophilicity? This will decide the place in the liposome. Then, once it occupies that space, or once it's designated a space, is that drug stable, or are there any additional considerations? For example, the oxidative stability of the drug, or does it hydrolyze, or does it have any instability factors, and these can be design considerations. The third factor is how the drug would affect the lipids, or the stability of the liposomes from the interaction with lipids. Now, if you have strongly ionizing drugs, which means extremes of pH, and then by extremes of pH, I mean anything outside of the let's say 5.5 to 8 range, then you must add buffers to maintain the pH. And so, yeah, these are the factors which whether drugs can have an influence on the phospholipids or the choice of the lipids.
What care should be taken in selection of the lipids for lyophilized products?
No specific care. All lipids, all well-produced lipids with are can be used for lyophilization. I would say it's not the choice of lipid or the selection of lipid which is a critical factor in lyophilization, but the process of lyophilization in itself. It's again a common mistake, because lyophilization is not unidirectional. You might pull out the water of the liposome and then you have no liposomes, you have only dry lipids and the drug in a solid matrix. So if you are using lyophilization, then you must know where to stop, the vacuum, the temperatures, and in general we would not recommend lyophilization except for very specific formulations. So there are easier techniques to dry out the the liposome, and they should be looked at first. Common technique, very easy technique, is spray drying under vacuum. It will cost a fraction, it's quite scalable, and you have no risk of damaging the liposome. So it's not so much the selection of lipids for lyophilized products, but the process of lyophilization in itself.
Okay. So, is there any ideal ratio for cholesterol and phospholipid in a particular liposomal formulation?
Yeah, it's almost like asking what should be the ideal wife. So it's the ideal answer to that question is it depends. Uh, there is no ideal ratio. The most common or wide range of ratio could be between 10 to 30%, but that's I know it's not saying, it's not answering anything. So as I said in the previous question, if you have no clue, then you could start at between 10 and 20% and then keep on optimizing in either direction, basically based on your desired release profile. uh, Good way is also to check using a DSC, differential scanning calorimetry, on what is the what is the phase transition overall you are achieving by this combination of cholesterol with the phospholipids. Then this factor is also influenced by which phospholipid. So if you are using, for example, if you used DSPC, which is fully saturated, quite a rigid, will always remain gel, then your requirement of cholesterol can be quite low. Except with if you are using DSPC and you want a 1 week release profile, then again your cholesterol content can go up. So yeah, there's no real ideal number. It's something you have to. I want to add here that when we talk of liposomes and lipids, and for different drugs and for different therapeutic areas and different sites of action, finally you get into a very precisely engineered nanoparticle. And trying to use thumb rules, trying to use standard lipids, standard formulas, standard ratios is probably a wrong approach. So we've seen greater success with researchers who work with an open mind, who will consider each factor independently, uh, go by a QBD approach rather than a standard approach.
Okay, we have a longer question, but then it seems it is what should be considered while making liposomes, the zeta potential or the liposomes, and in specific cases like using use of coagulates.
Yeah, it's indeed a long question, and I don't know if I can answer it completely in this short webinar. Uh, so just I'll take two parts. What is the relevance of the zeta potential? The zeta potential is, first of all, it applies only when your liposome is in an emulsion or a solution, and it's a stability of the emulsion or solution factor. Now if you had lyophilized liposomes, for example, then the zeta potential, or which is the difference of charge between the liposome and or the particle and the surrounding fluid, this becomes almost insignificant. The anionic charge on the phospholipid has a biochemical significance, so it will depend, or it will determine what is going to be the adhesion of the liposome to the specific target cell. So these are independent questions. Now, in case of cochleates, yes, absolutely. So you are adding or you are using negatively charged liposomes, and which will have a absolutely great effect on, or a greatly positive effect on the cell adhesion. The drug delivery across the cell membrane can be extremely high with the case of suitable charged liposomes in cochleates.
Okay. Uh, Again, about the phytosomes and liposomes, which grade of LECIVA would you recommend for making liposomes or phytosomes?
Yeah, first of all, there is to me there is no difference between a liposome and a phytosome. If a liposome is encapsulating a phytonutrient or a phytochemical derived molecule, it's a marketing term. Phytosome, it's essentially a liposome. You may see a series of words used to describe liposomes, ethosomes if it's ethanol containing, and so on and so forth. Now, how to select the grade is actually my entire presentation. So if you look at, for example, if it's a phytosome, you must look at natural versus compared to synthetics. Within natural, the choice is automatically plant origin compared to animal origin like egg. Within plant origin, you have the choice of soya bean and sunflower. If you can accept soya bean, that's the first choice. If But there are always concerns within the nutrition space for the GM non-GM question. We as a company, we always sell only non-GM phospholipids. And then the question on the allergen, which is more a labeling concern, or a label claim concern. If you want to make the claim free of allergens, then you can choose sunflower. Now, after having made this funnel, within sunflower, you will have 3 or 4 different grades. And I'm sure our marketing team is happy to connect and guide you with the choice within that narrow funnel of depending on your project.
Okay. So, if we use a mixture of synthetic lipids, is there any change in the phase transition temperature of the individual lipid?
No, individual lipids have phase transition temperatures as their physical property. So if you are using a mix, it is not altering the physical property of the individual component of the mix. So phase transition temperature is an individual lipid property. You will have a combination of phase transition temperature
Yeah.
lipids within the same liposome when you are using a mix. And so the release profile of the liposome is greatly influenced by the combination you use. But the simple answer is that if you use a mixture, then there is no change in PTT of an individual lipid. The combination is a clever design actually. I almost call it like, an artist will never paint with a single color but use a palette. A clever artist will use the palette very effectively, and you will get pieces of art.
Essentially. Is the size of the liposome depend on the nature of the phospholipid used?
Yes, but quite insignificant. The size of the liposome is, I would say, 95% dependent on the manufacturing technique. I am assuming you did not attend the previous seminar by Dominic Lipka, but I strongly recommend you ask for the recording or view the recording. And quite often, I mean, he's explained it in great detail. So the manufacturing technique, I might add, the wrong manufacturing technique can give you sizes which were not your target. You can get an entire mix or a salad of sizes. The dependence of the choice of phospholipid on the size, yes, it can happen, but for very, very specific projects. And even my question would be that if you had narrow polydispersity, why should it matter if your liposomes were 100 nanometers or 85 nanometers? It really doesn't, almost doesn't.
Okay. So, what is the role of cholesterol in the encapsulation efficiency of hydrophilic actives or drugs?
Absolutely none. So the role of cholesterol in encapsulation efficiency is absolutely none. The role of cholesterol in the release profile will be quite significant. So, let's say the shell or the membrane of the phospholipid or the liposome will start releasing as per its rigidity. And this rigidity is influenced by cholesterol. But the encapsulation efficiency is absolutely I would say, a manufacturing technique factor. And it could be also influenced by the choice of phospholipid, but not the role of cholesterol.
Okay. So, what are the stability studies conditions for liposomal formulations?
Oh, you can have everything under the sun. So you can have, I mean, practically you will normally not exceed the minus 18 limit, because finally you want to launch a formulation. You will not transport the formulation or store it or administer it at liquid nitrogen. So, but right from you can say the minus 18 frozen conditions, right up to room temperature, it's possible to design liposomes which are suitable for accelerated stability studies, uh, zone 4 studies. So it's a design factor. Actually, on the stability of liposomes, the choice of lipids is a critical factor. And if you chose a wrong lipid, then you can have disastrous stability challenges. And the problem is that if you, for example, if your liposome was designed to be stable under refrigeration, which means 2 to 8 degrees, there is no accelerated study possible which is extrapolable. Now, if you carried out long term studies at 2 to 8 degrees, and after 26 months you notice liposome failure, it can be a huge cost for your development project. I might add in a lighter vein that if you happen to be a junior scientist in the research department of a company, you may see your chair shaking. So it's extremely critical to start with the right design factors, because stability is the last mile on completion, and if you can lose a lot of hard work.
Yeah. Are there any factors like which will help to select edge activators or cryoprotectants based on the type of lipids?
Again, I mean, cryoprotectants, I'm assuming you are referring to the lyophilization process. First of all, we are not very big fans of lyophilization in itself. Then you may not actually need cryoprotectants. If your lyophilization conditions are designed to protect the liposomes, then they will not be extremes anyway. uh, and then the third part is that you can always add more gentle cryoprotectant. So our recommendation typically is minute quantities of sugars or carbohydrates, and if necessary, a small amount of glycerol. We don't recommend use of high quantities of cryoprotectants, simply because lyophilization is not the only choice of drying liposomes.
Okay. But the last question for today, would you recommend any combination of lecithins or lipids to target specific organs like heart or brain or any critical organs?
No. So there are no lecithins which can specifically target any organ, except then when you're targeting organs, again, it depends on the route of administration. I am assuming this is about parenteral delivery. So in parenteral delivery, you will have parenteral grades of phospholipids, and that's your choice. But within that, so once you chose a parenteral grade, then you must look at the other design considerations, such as the phase transition, the charge, etc.
Okay. So thank you, Mr. Kedia, for being with us today.
Pleasure.
And hope, yeah, to my attendees, hope we have contributed to give you the right direction towards your liposomal projects. After this, you would want to characterize or see the quality of your liposomes. So stay tuned with us for our next session. And do not forget to put your comments after the session. Thank you.
Thank you. Goodbye. And